everyone, and welcome back to ByHollyG, and welcome to today's video, which is going to be all about antibodies. The first thing I want to say before we dive into today's topic is that because of the situation that I'm in, I'm currently doing my master's and my research project. I'm quite busy at the moment, so I don't think I'm going to be able to upload a video every single week. So I hope you guys are okay with a video at least every other week for now, and then when I finish my master's towards the end of this year, I will be putting out a lot more content on this channel. But yeah, I just wanted to say that at the start because I know I have missed a few weeks here and there, and I've just figured that I think every other week is sustainable for me right now. But yeah, as you can basically tell from the title, in this video I want to share with you guys 10 facts about antibodies. Over the past year or so, going through the pandemic, we've heard a lot about antibodies. And so in this video, I hope that anyone, whether you've studied biology before or not, can gain an understanding of antibodies, what they are, what they do, why they're so important. So yeah, I hope you enjoyed this video. As usual, definitely give a thumbs up if you do. Feel free to comment down below any questions you have or any topic requests for future videos. And yeah, if you're new around here and you like the content on ByHollyG, biology content basically, then feel free to subscribe. And yeah, we will get straight into this video. The first question that we're going to answer is basically, what are antibodies? The first thing to say about antibodies is that they play fundamental roles in the immune system in protecting us against disease and, you know, fighting infection. More specifically, antibodies are soluble proteins that are secreted into the blood and other secretions, so like milk and sweat, for example. And they recognize very specific antigens. So we have a specific interaction between an antibody and an antigen. Based on what I've said so far, I want to address two things, just in case you were unfamiliar with what a protein is and what an antigen is. Proteins are basically chains of amino acids. A single chain of amino acids, a simple linear chain, is what we refer to as a polypeptide chain. And then this polypeptide chain can be folded and it's often modified, and that forms a protein. Sometimes proteins might be formed of lots of polypeptide chains that are folded and come together to form a complex. Antibodies are those types of proteins. They're made of multiple polypeptide chains, 4 polypeptide chains in particular. I'm going to talk a bit about antibody structure later, but that's a protein. Secondly, what's an antigen? So we know that an antibody binds to an antigen, but what's an antigen? An antigen can be defined as anything that triggers an immune response. Because people often get confused, what you can try and remember is that antibodies are the good guys and antigens are the bad guys. The antibodies are going to bind to antigens to get rid of those antigens. Now, antigens, as I said, they are basically anything that triggers an immune response. So that could be, for example, peanuts, because some people have an allergy to peanuts. But for now, if you don't really know much about immunity and antigens, then you can just think of an antigen as, say, for example, a microorganism that causes disease, which is what we call a pathogen. So for example, like COVID-19 will have antigens that are recognized by antibodies. Salmonella is another pathogen, a bacterial pathogen that causes food poisoning. You know, that is a pathogen. It has antigens on its surface. They are going to trigger an immune response, and part of that immune response involves the release and secretion of antibodies to fight those antigens and get rid of it. The next thing we're going to talk about is which cells secrete antibodies. As I said, antibodies, they are secreted into the blood mostly, but also into other secretions, and the cell type responsible for doing that is what we call a plasma cell. And plasma cells are specialized types of B cells, and B cells are immune cells that are part of the immune system. Adding a bit more detail, the immune system is broadly split into the non-specific or innate immune response and the specific adaptive immune response. And with antibodies and B cells and plasma cells, we're talking about that latter category, the adaptive or specific immune response. It recognizes specific pathogens. And when B cells specialize, they can become different cell types, one of which is a plasma cell. And as I said, that plasma cell is the cell type that is responsible for secreting those antibodies, which are those soluble proteins that we've talked about so far. Fact number 3, or question number 3, is what are immunoglobulins? If you have done any reading or research into immunology, you may have heard of the term immunoglobulin, and it's often used interchangeably with antibodies. They are subtly different, as I'm going to try and talk you through now, because there's quite a key concept that I want you to take away from this video. Put simply, antibodies are types of immunoglobulins, and immunoglobulins are the antigen recognition molecules of the immune response that are very important in the immune response. The word is so long and annoying, I can't even say it. I've said it already so many times. But anyway, immunoglobulins, right? They can come in two forms. They can either be a secreted form, and that's the antibody, or they can be a membrane bound form, and that membrane bound form is what we call the B cell receptor. So getting on to this important concept then. So as I said before, antibodies are made by a type of B cell called a plasma cell. However, before we get that plasma cell, we have to have a B cell that has a B cell receptor on its surface, and that B cell is going to be patrolling the body. It's basically circulating in the bloodstream and through the lymph nodes and the lymphatic system, and it's going to be just in search of any antigen that it recognizes. And immunoglobulins are very specific to a particular antigen. You can think of it like a lock and a key. A particular type of immunoglobulin will only recognize a particular antigen. If they fit together like a lock and a key, they have to be complementary, we say in biology, if they're going to fit together and bind to each other. But this B cell with a B cell receptor on its surface, that is the membrane bound immunoglobulin, is like circulating through the body, and if it finds an antigen and it encounters an antigen that it recognizes, that B cell with the B cell receptor stuck in its membrane is going to bind to the antigen, and that B cell, put simply, is going to become activated. And when it's activated, it divides and it differentiates into different types of more specialized B cells. One of those is the plasma cell, as I said, and that plasma cell is the antibody secreting machine. It's basically this cell that pumps out loads and loads of antibody. This plasma cell no longer has a B cell receptor on its surface. However, the most important thing that I want you to take away from this video is that the original B cell that had the B cell receptor on its surface and kind of like triggered this chain of events by recognizing the antigen, the specificity of that B cell receptor, i.e. the antigen, the particular antigen that it recognizes, that is exactly the same as the antibody that is eventually going to be released by the plasma cell. So for example, say if your original B cell recognizes an antigen on COVID-19, let's make it very relevant to the now. That B cell is going to be activated, it's going to divide and differentiate, and we're going to get this plasma cell that doesn't express a B cell receptor on its surface anymore. Instead, it releases the immunoglobulin in a soluble form, i.e. as an antibody. That antibody is also going to recognize the same antigen of COVID-19 that the original B cell receptor bound to. So as I said, they have the same specificity, they recognize the same antigen. In this way, we can therefore say that a B cell receptor is simply a membrane-bound form of an antibody, and on the other hand, an antibody is a soluble form of a B cell receptor. Now, it's not as simply as just like chopping off the B cell receptor from the surface and just like allowing it to be released as a secreted antibody. It's not that simple, but if you want to think of it that way to help you remember that, you know, a B cell receptor and an antibody, they have the same specificity if they've come from that same original B cell, then that's fine. And the final summary of this is that immunoglobulins, they are the antigen recognition molecules of the immune system. They can be a B cell receptor or they can be an antibody. B cell receptors are membrane bound, they're stuck on a cell's surface, and antibodies are soluble immunoglobulins. You can also try and say immunoglobulin really fast over and over again and see how you get on, and let me know down below because it's kind of a tongue twister. Immunoglobulin, whatever. Question number 4 is, how many antibodies are there in humans? As I said, an important concept is that immunoglobulins, antibodies or B cell receptors, they are specific to a particular antigen, and in humans our antibody repertoire is honestly huge. It's actually really crazy because we can basically generate or mount an antibody response to basically anything, because in the human antibody repertoire there are between 10 to the power of 9 and 10 to the power of 12 unique antibodies, and every single one of those will recognize a different antigen, and so it's therefore not difficult to understand or comprehend that, you know, we can basically mount an immune response to virtually any antigen. The big question then is that, you know, how are they all made? How do we make this diversity? Because the human genome only has about 20,000 genes roughly. We need to make 10 to the power of 9 up to 10 to the power of 12 different antibodies. How is that diversity, that huge diversity, generated? Now, I'm not going to go into the details of this video because it's very, very complex, but there are quite a few different mechanisms that result in the generation of this diversity. One of those is what we call VDJ gene rearrangement. As I said, I'm not going to talk about this, but that's just one of the many mechanisms that result in the generation of this huge antibody repertoire in humans. Also kind of related to that, question number 5 is, how do we make sure that we don't therefore mount an antibody response against ourselves? The answer to this resides in, again, a very complex process known as tolerance. Tolerance is a series of mechanisms and extensive processes that basically ensures that if an antibody recognizes a self cell or something that is harmless, then it's going to be removed from the antibody repertoire. So it's no longer gonna, you know, mount an immune response to a cell in our body or a peanut, for example, okay? There are many processes and mechanisms involved in tolerance. In biology, we never rely on a single mechanism because we want something to be robust. Like if you relied on just a single tolerance mechanism and that mechanism broke down, then you don't have a backup plan, you know? You have to have a backup of the backup of the backup plan, okay? But anyway, tolerance ensures that antibody responses are only going to be mounted when they're going to be helpful and when they're needed. Obviously, that doesn't always go to plan. That sometimes breaks down because, as I said, we do have autoimmunity, we do have allergies, etc. But tolerance, regardless, is extremely important. As with all of the topics that I've introduced so far on this channel, I am going to make lots more that go into more detail. Even with antibodies, for example, there's so much more I could say. But the next question that I'm going to talk a bit about is the structure of an antibody. So what is the structure of an antibody? As I said at the very start, antibodies are proteins. And if you also remember, I said that antibodies are proteins made of more than one polypeptide chain. And remember that a polypeptide chain is just a linear chain of amino acids that will be folded in a certain way to make a whole protein or part of a protein. In this case of antibodies, polypeptide chains will make part of a protein because antibodies are composed of 4 polypeptide chains. They look like a Y shape, so we describe them as Y-shaped proteins, and they are composed of 4 polypeptide chains. We have 2 heavy chains, so a pair of heavy chains, and 2 light chains, so a pair of light chains. It's a symmetrical structure. If we drew a line down the middle, you'd have 1 heavy chain and a light chain on one side, and then a heavy chain and a light chain on the other side. Each of those 4 chains is a polypeptide chain, and they're held together by various bonds. And the antibody is also quite a flexible structure because the antibody needs to be able to bend and contort itself so that it can interact with antigens or an antigen. Without going into too much detail, I want you to know that every single antibody has 2 antigen binding sites. So an antibody can bind to a maximum of 2 antigens at a time, and as I said, because antibodies are flexible, that allows them to bend themselves in a way that allows them to bind to a maximum of 2 antigens at a time. Every single polypeptide chain of an antibody, whether it's a heavy chain or a light chain, they have variable regions and they have constant regions. Now the constant regions of antibodies are constant, hence the name, they stay the same, whereas the variable regions, they are variable. Those are the portions of antibodies that will change and determine the specificity of that antibody. So it will determine which antigen it binds to. And the antigen binding sites of an antibody, which are labeled on this diagram, they are composed of the variable regions of the heavy chain and the light chain. So the variable domains of the heavy and light chain come together, and that forms a single antigen binding site. So as you can see on this diagram, we therefore have 2 antigen binding sites in a single antibody, in a single Y-shaped protein. Question number 7 is, what are the different types of antibody? Because we have different types of antibody, we call them different isotypes. And there are 5 main classes of antibody, and the class or the isotype of antibody is basically determined by the heavy chain. They are IgA, so Ig stands for immunoglobulin. So we have IgA, IgG, IgM, IgD, and IgE. And as I said, the letter is basically coming from the type of heavy chain that we have in the antibody. For example, IgA has an alpha heavy chain, and so that's why we call it IgA with a capital A. And so like IgG, for example, the heavy chain is the gamma chain, so it's IgG. And basically the isotype of the antibody, or the heavy chain that we have in the antibody, that determines the particular function of the antibody, what it can kind of do in the immune response. We actually also have subclasses within those 5 main classes. So for example, we have IgG 1, 2, 3, and 4. Before we move on to question number 9, which is basically what are the functions of antibodies, I want to firstly address how do antibodies exert a particular function, because I think this is important to talk about before we discuss the actual functions. Now, we can say that antibodies have 2 functions that are spatially separated. Now, the first function is antigen recognition. Via the antigen binding sites, an antibody is able to bind to a particular antigen, and that antigen will be complementary to the antibody. As I said, they are specific to each other. They bind like a lock and a key. So whilst the antigen binding site provides the antibody with specificity, the constant regions of the antibody that I'm going to label on this diagram, so the constant region of that Y-shaped protein, that region of the antibody is responsible for interacting with other cells and other molecules in the immune response that allows the antibody to exert a particular function. So it therefore translates that antigen binding into a useful function, so the antibody can actually do something. If an antibody can bind to an antigen, that's fine, but we want it to then do something, and that's what the constant domain is responsible for. You know, it interacts with other parts of the immune system and it allows the antibody to perform a particular function, and those functions are now what we're going to turn to talk about in this video. So there are loads of functions of antibodies, but I wanted to talk about the key functions of antibodies. So what are the key functions of antibodies? So the first function of an antibody is basically something that we call opsonization. So this is a key word in this video. It sounds quite complex and confusing, but trust me, it's not that bad. Opsonization is basically the process of flagging a pathogen so it is then removed. So what an antibody does is it will bind to an antigen, as I said, via its antigen binding site, and that kind of acts like a flag. Okay, if you imagine a ship that is out at sea and it's stranded, it puts up like a red flag or something. That is a signal that it needs help. This is like what the antibody is doing in opsonization. You know, it binds to the antigen on the pathogen, on COVID-19 for example, and that acts like a flag. That is opsonization. It's flagging to the immune system that this antigen needs to be removed. And so once we've labeled or flagged that antigen, other cells of the immune response will come along and they will remove that antigen. So that is the process of opsonization. It's labeling or flagging an antigen so that it is subsequently removed by the immune response, and it's removed in a process called phagocytosis. We have these cells in the immune system that come along and basically engulf pathogens and antigens. So the phagocytes will recognize these antibodies labeling the antigen, and then via the constant domain, as I said, on the antibody, phagocytosis will take place and the antigen will be cleared. The second function is what we call ADCC. ADCC basically stands for antibody-dependent cellular cytotoxicity, or antibody-dependent cell-mediated cytotoxicity. So an antibody, it may bind to an antigen on a cell that needs to be removed. So for example, like a pathogen, for example, a particular bacterial invader. After that antibody is bound to the antigen on that cell that needs to be removed, that cell will be killed, and that's what cytotoxicity means. Cyto meaning cell and toxicity meaning toxicity, so it's going to be killed basically. Antibodies are basically facilitating killing of that cell to which it is binding to. Again, we see that the antibody has this spatial separation of antigen binding by the antigen recognition domain and then the function that is exerted. So in this case it's killing, cytotoxicity. Other things that antibodies can do, they can basically neutralize toxins. They can also be responsible for triggering the release of further inflammatory molecules in the immune response to heighten the overall immune response against a particular invading pathogen, for example. They also can perform a process called agglutination. So antibodies will basically clump the antigens or the pathogens together, and we form this big clump of antigen antibody complexes. And if you form this kind of clump, it's much easier to remove a single clump instead of the immune system going around to try and kill every single antigen or pathogen individually. So it's basically more efficient for the immune system. So if you form this big clump, that can simply be removed all in one go, and that is, as I said, agglutination. You're basically like gluing or sticking things together in a big clump. The final thing I did want to mention under this topic of antibody functions is that they can also be transported in the body. So for example, IgA, so the isotype IgA, that is transported to epithelial surfaces. So one, for example, is the gut. So IgA will be secreted into the gut lumen. IgA is also transported into milk, so for example like breast milk. And IgG, so another isotype, that is transported across the placenta, so from the mother's bloodstream and into the developing fetus, and that provides the developing fetus with immune protection early in its life, early in development basically. So the final question, question number 10, is basically how are antibody responses terminated? Because obviously a very important part of the immune response not only involves generating a robust immune response, but it also involves dampening it back down so we don't have inflammation in our bodies running wild like wildfire, and so we have to be able to dampen down or get rid of those antibodies. Antibodies obviously have a particular half-life, they will eventually break down, but antibodies are very clever. They basically set up their own negative feedback loop, so the antibody response will be terminated once it has been generated, and that will mean that the antibody response dampens down, we restore normal conditions, we don't have any inflammation anymore, and that makes sure that, as I said, there aren't any detrimental consequences to overinflammation or chronic long-term inflammation and stuff like that. So those are your 10 pieces of information, 10 nuggets of information about antibodies. As I said at the start, I hope that through these 10 facts or 10 questions being answered, you better understand antibodies, or you've been introduced to the concept of antibodies. They also have so many other applications, not just to do with the immune response inside our bodies, but they're used extensively in biology research, and yeah, they're very, very powerful molecules. So yeah, I hope you enjoyed this video and learned something from it as usual. Definitely give this video a thumbs up, as I said at the start, if you enjoyed it. Comment down below, subscribe to join the ByHollyG community, and I will speak to you very soon in another video.